Lara Ray talked about a clinical neuroscience approach to medication development. Medications development for addiction. Thank you, Lara. Mark?
Thank you so much for having me. It's really a pleasure to be here and to learn so much about ways in which what we do impacts the community. What I want to do, based on what I've been learning this morning, is just to really keep my presentation short and maybe have a little bit more time for discussion. So I won't cover nearly as much of what I intended to cover, but I hope what I can do is give you an example of how some of the basic clinical neuroscience approach can actually come, hopefully, all the way to informing new treatments. And so I'm a clinical psychologist by training, and a lot of my research has been integrating these tools or methods, including genetics, pharmacology, neuroimaging. So obviously the methods represent some of the ways in which we can get at psychological and phenomena. But I argue that a clinical neuroscience approach is less about the methods and more about the thinking and the theories behind why some of the human and clinical studies are designed. If we're really thinking from the basic science all the way to clinical science with humans, the question is less to me about which methods are being used and more about the hypothesis generating, hypothesis testing that is really bridging between what we know in animal models and what we can learn and ultimately impact lives by testing human subjects. My work is primarily experimental. So I have clinical trials that I'll be telling you about, but it's really about bringing in and looking at some of the mechanisms that may predict response to various treatments. So I'm going to focus on sort of a broad slide of things that I've been doing in my lab, and of course I could spend a whole lot of time on this. I'm going to focus on really, I thought about you, but I'm going to focus only on one of these areas of inquiry, which is the development of a new medication, a combination of pharmacotherapy for smokers who also drink heavily. And I have some interesting work on methamphetamine, but I will leave that for another opportunity. You can always email me. I'm happy to share papers and slides and so forth. So this is actually one of my first grants when I came to UCLA, and it's a TRDRP grant. It really got me started with a new investigator TRDRP grant funded in 2009 that I ran from 2009 to 2012. And this grant really looked at the simple question of, we know that smoking and drinking go together like, right? We know that folks who drink like to smoke, and drinkers are more likely to be smokers than non-drinkers. So one of the questions is, well, what is the implication of this if you have a patient who's trying to quit smoking, but also happens to drink heavily? Well, what the research says that, lo and behold, these folks are going to be much more likely to relapse. So if you're counseling someone who's a smoking patient, and you know that your patient is a heavy drinker. Now, heavy drinker doesn't mean dependent on alcohol or heavy in alcohol use disorder. Heavy drinker is defined by National Institute of Alcohol Abuse and Alcohol. Really, someone who has 7 or more drinks per week, so a woman, and 14 plus for a man. So right off the bat, we're talking about 20 to 30% of all smokers. This is not a tiny subset of smokers. It's about 20 to 30% of all smokers. So now I'm bringing you to your patient who is a smoker, wants to quit, and is also a drinker by this definition. Well, guess what? They're 5 times more likely to relapse during a drinking episode than collapse occurring outside of the episode. So now we have mechanistic data to say, look, drinking and smoking goes together, and drinking poses an obstacle to smoking cessation. And more importantly, this is a sizable subset of smokers that we need to treat. So my question is very simple. We have varenicline, which is Chantix. You're familiar with it. arguably the field standard for sedation, a very good medication, partial agonist effect of the nicotinic acetylcholine receptors, a very good medication, right? And we have naltrexone, which is a medication with modest effect sizes for reducing drinking. The question is, would the combination of these two medications be superior to monotherapy for these heavy drinking smokers? Now, before testing this in a clinical setting, what I'm going to do is show you results from my TRDRP study, which was basically a mechanistic study. It was a study that brought people into the laboratory and assigned them to one of these conditions. These folks would get either naltrexone alone, varenicline alone, or their combination. They could also be assigned to placebo. So effectively, we have 4 conditions: placebo, the 2 monotherapies, and the combination pharmacotherapy. And we randomized 130 individuals, 120 of them completed. So we had 30 individuals who were heavy drinking smokers in each of these conditions. And now we're asking them, we're bringing them to the laboratory. They're abstinent for 12 hours. They're very grumpy, very uncomfortable. They're really looking forward to having their first cigarette. And, I have all these really lovely participants at UCLA, and they're getting treated. These are very grumpy smokers that are coming in. So we know that that's part of the withdrawal syndrome, right? You ask them to stay for 12 hours, they get pretty grouchy. So what we do is we assess them when they come in. We assess them after giving them alcohol, because again, there's this alcohol-smoking interaction, so we give them alcohol, and then we test them again after they smoke the first cigarette of the day. So here's what we find. We did not find significant differences at baseline when they first came into the laboratory. So what we find is that after giving them alcohol, you ask them about their ability to resist smoking, and this group, the varenicline naltrexone group, the combination group, did better than monotherapy and placebo in terms of their self-report of how capable are you of resisting the urge to smoke right now. When we ask about how much they enjoy the alcohol, they also seem to report less of a subjective reward from drinking. Now remember, their abstinence, we verify their abstinence by getting a CO reading, and now we say, okay, well, here's your first cigarette. Let's have your first cigarette of the day, and we repeated assessment. What we found, again, is the combination of superior to placebo. The monotherapies were not superior to placebo in terms of reducing craving for the next cigarette. And also, we found a reduction in terms of their high, how much they reported enjoying that first cigarette of the day, which is very critical for dependence. This is an unexpected finding. So what I'm about to show you is an unexpected finding. We have to titrate drinking for a period of about 10 days. Okay? So we did not ask anyone to reduce their drinking or their smoking, so we kept interviews of how much they were drinking and smoking as the medications were being ramped up. And what we found, if you look at drinks per drinking day, is a general reduction on the combination group and also on naltrexone. So maybe naltrexone was driving some of those reductions in drinking. And interestingly enough, folks started reducing their cigarette smoking, and more so in the varenicline naltrexone condition. They reduced it by 2 cigarettes. So they weren't quitting, but naturally, without us prompting them to necessarily quit, they were reducing their smoking, which was not part of the aims for a TRDRP proposal, but I think it gave us a lot of ecological validity in terms of, there might be something going on here with people's not only subjective reports, but also what they're doing with their smoking and drinking behavior. So, of course, we're interested in moving forward, perhaps with the clinical trial, as Dr. Branch discussed elegantly this morning. There's a lot that we can learn from the neuroscience approach and understanding the brain in ways that can inform treatment. And so I was fortunate to collaborate with Dr. London, Dr. Lemay here, and to really develop a study in which we have a subset of folks for this trial come in and do a neuroimaging task for us. So what we do is we have videos of people doing everyday things like And we were interested in saying, well, how does this combination pharmacotherapy work in terms of attenuating Q reactivity? Is it superior to placebo? Is it superior to monotherapy? Now, we have some evidence, I'm just reviewing some of the research here, suggest that varenicline would reduce orbitofrontal cortex activation during the cigarette cues compared to controls. We knew less about naltrexone. So we had a subset of 40 individuals, 10 in each group, and here is the pattern of activation that we found. So essentially, we compare how much of a bold signal response, oxygenation response we got from cues versus neutral images, just the newspaper without the cigarette. What you find is pretty wide activation in areas that would expect to be implicated in addiction. So you see amygdala, insula, anterior cingula, areas that were highlighted earlier. Again, the consistency across studies is a different question, but we were encouraged to see this level of activation of cues. Now the question is, well, how do we do as far as medications? Well, if you look here, again, we have varenicline alone, naltrexone alone, the combination of placebo. You can see that for the last nucleus accumbens, all active treatment conditions attenuated activation of these brain, last nucleus accumbens compared to placebo. So here we didn't differentiate much across the active medications versus placebo. When you look at FFG, we found the difference between placebo and the combination. What was most striking to us is really looking at the anterior cingulate. Here you find that the only condition that differentiated from placebo and also from the monotherapies was really the varenicline naltrexone condition. So this was exciting to us and really indicated that this combination may be promising on a number of levels. So from the neuroimaging data to the human laboratory data to the reductions in smoking during the titration period. We looked at logography, which is another measure of urgency to smoke. We record their smoking behavior. This really came together nicely for us in terms of the picture that this combination may be useful. And luckily for us, neither of these, and we are now on the third year of a large research grant from R01 that is testing the combination of naltrexone versus varenicline alone. So the idea here is that we have a standard of care. We've worked very hard and invested a lot of research dollars in developing varenicline. And the question is, can we do better by our smokers who are also drinkers when we add naltrexone? That's a critical question. Now, of course, this is a superiority trial. And what that means is that you're really trying to improve upon a treatment that's already pretty good. And also what that means is you have to really recruit a lot of people. So the last thing I'm going to tell you is kind of put a pitch for you to really consider referring patients to our study. Everyone needs active treatment. So we know that the two conditions are, Chantix alone, very good treatment, Chantix plus naltrexone. Okay, so everyone receives physical, everyone receives counseling, everyone receives medication and compensation. So we're really working on a lot of socially, economically disadvantaged participants, folks who are really engaging with the UCLA healthcare system to start smoking. And we've been really encouraged. We already randomized 100 subjects, which is great. And we have another 100 studies to go. So if you're interested in perhaps one of the other studies, please send me an email. You have my contact, and I'd love to talk more about flyers, brochures, book parts, so we can get this information out to the public. We also pay for travel. I understand, of course, traveling in LA is expensive and time-consuming, so we're more than willing to support our participants in travel costs as well. So I'm excited because I'm happy to see this work progress from the lab to the clinic. I'm also excited because we get to, see people quit, which I tell you is a lot more fun than making them abstain when they don't want to and they're really grouchy. These folks are quitting, and it's really rewarding to see. What would be even more rewarding would be to have a new standard, and that's what I tell my staff all the time. We're going to randomize 272, not 271, because we really need to know the answer to this question. We have the potential to have a really important impact in the field, and we're committed to that. So I would love to add a neuroimaging component to working hard to identify, to follow up on my my finding over here, the AC6 signal as a potential predictor of response. I think that would be super exciting, in line with some of the NIDA priorities, and something that could be a biomarker of response. So that's pretty much all I want to say. I want to recognize friends, collaborators, students, and like I said, I'm happy to share more on our work on my lab website. You're welcome to email anytime, especially when I acknowledge Lara, who's here, Edu London, some of the collaborators on this work. And obviously here, and NIDA for their support as well. So thanks, everyone.
I'll start with a quick question. Do you think they're going to develop a pill that's going to combine ranitidine with naltrexone? Do you think a pharmaceutical company might develop a pill that combines the two in one?
Potentially. I mean, we do collaborate with pharmaceutical companies. I can't pretend to understand all of their financial interests. It's beyond my comprehension. I would be happy enough if physicians are willing to prescribe both as a starting point, if we felt like we had enough data to suggest that is an improvement upon the field standard.
I'm really struck by the heavy drinking definition, especially given data showing that 1 drink a day may be good for our health. In fact, I did like an Alzheimer's prevention with my phone and told me to drink more. And so, and also, also smokers are going to be high risk for cardiovascular disease. So you comment a little bit more on that particular section.
Sure. So this is a widely applied definition that comes from NIAAA. There is an element, we use the 714 number. You can add a binge drinking element to it, which some studies do. They say you have to have 8 binge drinking episodes. My perspective from working with these smokers is that if you think about 1 glass of wine a day, start to get paired with that 1 cigarette after a meal with a glass of wine, and that's enough to create enough sufficient association that it's hard to disrupt. Does that make sense? So I see your point about that's a low threshold for drinking, and I think for the purpose of understanding the connection between drinking and smoking, we have enough of that sort of daily habitual pairing of the two. That might be what's clinically. But yeah, I think a lot of people have a strong reaction to the definition.
Sure.
Is there any difficulty in taking both drugs at the same time? Are there any side effects? Are there any drawbacks to doing that as a clinical or something?
So essentially, in our care, we have a low dose of naltrexone, 25 milligrams, and then through the review cycle at NIH, it went back to the 50 milligram dose. Interestingly, and I'm not a prescriber, but what I see my patients for in this study is that if you have headache, which you need to titrate varenicline, that gives you a really nice window to also titrate up naltrexone. So you can be very gentle in titrating naltrexone, and people tolerate it really well. So we have not had any issues with dose reductions or significant dropouts. If people get to those 10 days, really gentle with naltrexone, which has a primary side effect of nausea. You can also use injectable naltrexone, but there are a lot of issues with pain management, there can be vernacular, there are all these things that you will put yourself up to if you do kind of different dosing.
Great job. Right on time.
Thank you. And I might need to type LA traffic, so I apologize if I don't see some of you. Please do email.
Thank you so much. Testing.